Good morning, everyone. My name is Dr. David Guiliano. I'm the head of Department of Biosciences here at the University of East London. And today we're going to have a roundtable discussion with some of a good collection of my colleagues about COVID-19 and how COVID-19 has driven innovation in the biological and chemical sciences and what opportunities this has created for science graduates. So what we're going to do is we're going to go through and talk about a couple of key topic areas and then take the subjects back to what is happening in terms of development in these key industries and scientific areas, and then talk about what opportunities this is creating for graduates. So hopefully, as you're thinking about your own career, either starting a degree in one of the sciences or finishing your degree in one of the sciences, you'll see how you could potentially go in and explore some of these subject areas as part of your career. I'm going to just start by introducing my colleagues. I'm going to introduce Professor Mike Seed. Hello there. Professor Seed is Professor Emeritus in Pharmacology in the department. I'm going to introduce Dr. Winston Morgan, who's Reader in Toxicology, and Professor Sally Cutler, who's Professor in Medical Microbiology. Welcome, everybody. I'm going to start with Professor Cutler, and I'm going to sort of put out a very clear question about the NHS clinical science services because they were so visible in our response to COVID very early on. So, Sally, can you tell us about how the NHS clinical science services have had to respond at an extremely rapid pace to the spread of COVID-19, and how that's had to then be sort of integrated into the national response to the pandemic? How has this driven innovation in the clinical sciences?
Okay, thank you for that. Well, I think the initial thing, as you said, they had to be early responders. They were dealing with patients, they needed diagnostics rapidly. And so the first thing that actually happened was the staff had to be, a lot of them were redeployed from different areas of their diagnostic biomedical scientist roles. And they had to come and support the diagnostic virology services. And so this actually really sort of shows the value of things like the broad reach of that background people get, certainly during their training at university in biomedical sciences, because those skills that they had that maybe they hadn't used for a few years had to rapidly be deployed. So the agility of staff was really, really key to get that really good early capacity to accommodate the rapid diagnostic numbers that people had to cope with. So that was one of the first things that actually came through. We also saw some quite innovative practices as well, such as answering a lot of the diagnostic questions and challenges that people had, in the fact that they didn't know which assay was going to be best. There were lots of discussions about that. And so we actually saw a really good innovation come in there where people were using a crowdsourcing approach. And so they set up a web page and they said, hey, well, we don't know what sample type is the best one to use. Obviously, to get a really good bronchial lavage sample or something, a nasopharyngeal swab is great, and that might be the ideal sample. But then you also have higher risks in taking those kind of samples. And so obviously it's more uncomfortable for the patient. And so it was, how do we do this? How do we get around this challenge? And so people were able to post ideas, run little trials and say, well, we looked at this and we found actually dry swabs work better than these other swabs. Viral transport medium wasn't needed. Actually, you can use other approaches. And so a lot of progress was made that way. So that was really exciting as well, to see how people were working together rather than competing. And so I think that was a very good example of innovation. There was also a rapid scaling up of diagnostic capacity. So we saw things like the Lighthouse laboratories that were really good for high throughput testing. because that's what the country needed, and they needed them really, really quickly. And we actually saw a lot of our graduates going and helping support the capacity in those laboratories. So I know quite a few of our students have gone and helped on the front, really, sort of doing those diagnostic tests. So we've also seen lots of rapid research that's been done to try and get a better diagnostic test, which PCR, which primer sets. Different countries around the world were proposing use of different ones. We've seen comparative trials, and we've actually seen sort of best practice come to the forefront, which was really exciting to see. A lot of effort has also gone into testing the various antibody tests. A lot of the early tests that were out there were really unreliable. And so we didn't know that until those validations were actually done and they were run in parallel with each other. So again, we're seeing refinement of the diagnostic tests for antibody testing as well. And only today we're seeing now a UK-based firm, Oxford Nanopore, jumping in on the initiative for really sort of getting a rapid diagnostic test, because these other tests that I've mentioned, the PCR-based tests, the antibody tests, they take time. They'll take several hours to run. And when you've got a high throughput diagnostic facility, sometimes people are having to wait days to actually get the results. You need the result today. You need to know now. You've got somebody who's feverish, you think they've got COVID-19, you need your answer now. And so the Oxford Nanopore system, LAMPOR, is actually giving it a predicted 90-minute turnaround. It also doesn't need the technical skill base that a lot of these other assays actually need. So you can actually take the test to the patient, and you can get somebody who hasn't really got a huge expertise in doing a lot of these other types of tests to actually deliver the test and get the results. And not only will it tell you whether you've got COVID-19, but you can actually expand this to look at other things that might have similar clinical presentations and might be confused with COVID-19. So you can do a full respiratory screen as well. So we're seeing all of these really exciting, innovative practices coming to the forefront, which I think it's really a great time to be involved in the field.
I think particularly that point of care diagnostic development is one of the key things that is going to be the areas of big development in the coming months. Winston, Mike, was there anything you'd like to add to what Sally said?
I'll let you go, Winston.
No, I was going to say I was going to move into my interest, because as I've been watching this process take place, you become interested. At least I became interested in the outcomes. Who is affected? Who's not affected? Because unless you know who's affected, who's not affected, only then can you decide what the treatment's going to be, who needs to be protected. And very early on in the pandemic, we noticed, for example, that older people were affected. People who are 65. I think it's about over 95% of all the deaths have been over 65. So clearly, very early on, we noticed that people who were older were more susceptible. And then we looked a bit closer. We noticed that, for example, you needed to have underlying medical conditions, for example, high blood pressure, obesity, diabetes. These were very important. So it's a combination of a number of factors, age as well as underlying medical conditions. One of the, if you like, a bit of misinformation. People thought that younger people weren't infected, but they were probably infected but not affected, partly because early on in the pandemic, Sally was talking about the diagnostic testing. One of the things that you could only afford to test people who were showing symptoms. So younger people, for example, were asymptomatic, so we didn't need to test them, so they could have been infected. So we didn't notice that. So having seen that, if you like, older people were affected, people with certain underlying medical conditions were affected, people then thought, what's behind this? And the most obvious place to look are genetic factors. Are there any sort of genetic factors that might lead to this? And there have been a number of studies, not many clear-cut. Perhaps the one that has been most clear-cut is blood groups. For example, we notice that people with type A blood groups are just slightly more likely to be affected, whereas people with type O are less affected. There have been other areas where genes may play a role, and it may come into something that Mike wants to talk about in terms of drug treatment. So if you express high levels of a receptor called the ACE2 receptor, if you have high levels of that, perhaps in your lung, in your kidney, that kind of thing, you're more likely to be infected by the virus. And one of the ways you can increase your expression of this receptor is by taking certain drugs, for example, drugs to treat high blood pressure. So having high blood pressure could be the reason why perhaps some people are more infected. There are lots of other factors. For example, people talked about different ethnic groups. The problem with that is that ethnic groups and race go across, they have intersections. So if you think of, for example, Indians and Pakistanis, for example, they're both in the same racial group, but in different ethnic groups. And you were seeing different outcomes for the two different groups. So it's for scientists to think about how you look at data and how you analyze data. So that's brought in a lot of innovation. So senior scientists are having to look again at how they analyze data. Do we need to change the categories? Because a lot of these categories are produced by the Office for National Statistics. And they've traditionally categorized disease, if you like, outcomes in one way. What COVID has done is to say, no, we really need to look again and see if there are different ways we can categorize people from different groups, because it will tell you so much more. I think I'll stop there for now.
Thank you, Winston. But I think I want to come back to you and ask. So, what sort of opportunities do you see for instance, young science graduates to sort of in this area? Because it sounds very exciting. This intersectionality of study and of thinking, which we now need to do when we're looking at this data to try and understand it properly. What do you see as some of the key opportunities for the next generation of scientists to kind of contribute to this?
Well, if you're a scientist, traditionally, as I said, you basically just learn the science, learn the biochemistry, the biomedical science, the genetics, the pharmacology. You've just learned that. And you haven't really looked beyond that in terms of the if you like, some of the social factors and how these play out. So what I think scientists in the future are going to have to do is not only learn the genetics, but how does the genetics play out in wider society? And that's going to be quite important. The other sort of, if you like, opportunities and innovations, I think, is for scientists is that we've got lots of drugs, and I'm in a sense stepping on some of Mike's territory, but as a toxicologist, I think I can do that. Lots of drugs on the market that we've used for one thing, and suddenly we're thinking, could we use it for something else? Because that's what some of the things that we've discovered, that drugs that were discovered for one condition may actually be effective against the virus. At least that's what people think. Or it could be around the concentrations of those drugs or how they're used. So there are lots of opportunities that biomedical science students, pharmacology students, pharmaceutical science students will have. COVID has, if you like, opened up our eyes in a way that COVID has sort of shone a light on science and how we use science in a way that it never had before, and just shown society how important science is and how important scientists are.
Thank you.
I think also, just to interject, it's the multi-skilled nature of it, and transferable skills. So the biosciences are probably the most sort of intersectional graduates that are produced, because they cross over from chemistry, genetics, the cellular organism, the whole organism, and across a whole wide variety of sciences. So it's the application of a lot of sciences. So, we are, as bioscientists, we are highly employable in this regard.
So that leads me to you, Mike, and the question I sort of had for you, which is based around what everyone says we need, which is this acute sort of need for effective treatments for people who are suffering serious infection with COVID-19. How has industry and the regulatory authorities responded to this need?
This is amazing, because it started off basically as a Wild West. Everybody trying their own pet thing, and of course China was at the lead, preprints from different groups suggesting on small patient numbers that this drug, like hydroxychloroquine, was working. Azithromycin was another one. In the UK, we're very lucky that we've got a National Health Service, and very quickly, within 2 weeks, 3 weeks, they set up 2 platform clinical trial bases. One called the Accord, which was for early phase experimental drugs, and the Recover, which was for the repurposing of existing therapies, but also taking drugs from the Accord trial as well. And basically every patient who came into the NHS was asked would they like to join. So that's 22,000, or they've got something like 27,000 people on this, and being a platform, they can test different things and can stop different things as well. And this is on a pure hard-nosed clinical trial driven basis. The regulatory authorities have allowed this incredibly quickly. They've almost waived the ethical process. So we're leaning on our past experience with this. So for the Recover, for example, so out of China, we had the hydroxychloroquine, ponavir, ritonavir. Those are antiviral agents. The anti-IL-6. All of these came out of the small trials. In this large Recover program, those are found to be ineffective at reducing the more serious cases. So that's really important. Because we can concentrate on other things which aren't going to work. So low dose dexamethasone, that was a really clear signal. But it's not just this organized area. The other areas, people are still trying things in the clinic that they think will work. So Bradford, for example, they're using what's called CPAP, which is continuous positive airway pressure. They have introduced that really early on. By doing so, their ICU admissions are 50% of the norm. So then that was just them trying it in their own NHS area, using their own expertise. So that is now entering proper clinical perspective trial as well. And there are other areas now. The drug industry are repurposing some anti-IL-33, for example. That's an amazing. It looks, well, personally, I think it could be an amazing success. But they've also knocked their own drugs on the head, like the anti-IL-6, for example. Roche, their clinical trial has shown it to be ineffective. The UK, we're not so sure, but it's included still in the Recover trial. They still think it's got hope, so they haven't withdrawn it yet. Okay. So it's really exciting, really fast moving,
very dynamic. It's a very dynamic playing field.
Yeah, what we're seeing is that both the regulatory authorities and the scientists have really sort of become very flexible in how they work, and very maneuverable. They haven't just sort of just stuck to what they've done in the past. They've seen the challenge and the dangers of COVID-19 and the need for new therapeutics, and they've responded, and that's really important. And that's what when we train scientists, you train to be flexible, you train to respond to the world around you. So you have to be able to see things and respond to it. You don't just stick to one thing. That's the benefit of being a scientist.
I think it's very important to make sure that the educational message gets out there as well, in the fact that you need to know which of these therapeutic interventions is going to be appropriate to use at which stage of the infection. Because obviously antivirals, you should be using those at the very early stages. To try and prevent the legacy, the more serious complications that might happen later. Those that target the immune system, the interventions that you outlined and discussed there, they will probably play a bigger role once people are beginning to develop more serious complications. So I think we need a lot of education of our medical colleagues as well of what to use when. Get the prudence.
Mike, can I just ask, what do you see as the big opportunities for future graduates, these people who are coming out of their degrees now in sort of drug, in these areas that might contribute to drug development or clinical trial oversight? What do you see as some of being the key opportunities?
It's fantastic. So we've got the normal drug discovery area. That's showed everybody how important it is. So I can see more investment going into drug discovery. But if we're talking about clinical sciences and clinical trials, it's not just nurses and doctors who are involved in this. There's a whole team behind them. So we've got management, data collection, we've got the analysis, the people in the labs involved with it, but it's the auditing as well, identifying toxicity signals. You need to be understand how drugs work, how biological systems react. We need to understand the importance of audit, of reporting, and regulation. And our graduates, for example, go into all of those things. It's a very wide area. As long as one doesn't get fixated on one area, I must work in the lab on the genomics of this. Suddenly realize your skills are very diverse and highly desirable.
I think I was just going to say, one of the important areas is people scramble for new therapeutics, because although a lot of the talk is about the vaccine, there are questions about how effective it's going to be, is the fact that you need to design new drugs. And our graduates, especially the pharmaceutical science graduates, they get well trained in using computer models to design new therapeutics. And so you get your hands on lots of software designing drugs to fit receptors and that kind of thing. So it's a really important thing that we do, and you learn as part of your skills, not just laboratory, but also using computer models to design drugs to fit receptors or targets.
In silico science.
Yes.
I mean,
I mentioned a key
graduates.
Thing, Winston.
Sorry?
You mentioned a key point that we haven't really discussed yet, and that was about vaccines. So I think looking to the future, prevention is better than cure. So this is more your area, Dave. Just bring us up to date on what's happening with vaccines. And
so, I mean, like the drug development and repurposing field, the COVID pandemic has opened up a lot of possibilities, and so a number of different vaccine technologies have been applied now to try and develop an anti-COVID-19 vaccine, and some of those have already gone into phase 1 clinical trials. A lot of that has been very publicly sort of disseminated. I was just looking on the BBC, and every day you go on, every couple of days, there's an article about either the Oxford vaccine or the Imperial vaccine or one of the commercial vaccines which are currently being developed. Like the drug development and repurposing, it's the acceleration of the application that is really, really different and exciting. And it means that technologies that we've sort of been developing quietly in the background are now being given the chance to sort of be tested out fully. And that's great, but it also comes with a caveat of we have to make sure we are respecting, as Mike said, due diligence for the people who are being given these potential new vaccines, and making sure that they don't pose any health risks. And so I think it's very early days. I mean, as I said, every couple days you turn on the BBC and there's something about how we're going to have a vaccine in 6 to 8 months, 6 to 12 months. I'm a little bit more reserved in my judgment on this, because one of the things that's really different about vaccines, in terms of the safety hurdles that you have to pass to actually have a usable vaccine, it's much, much higher than it is for some of the drugs, because basically what you have to show is that when you give somebody a vaccine, one, it's effective, great, but also the health risks of giving that vaccine are much, 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 much lower than someone might encounter when if they were to get the disease itself. And because of the heterogeneity of COVID, and what it does to people, I think that's going to be an incredibly difficult playing field to sort of work out. You're essentially going to have something as safe as one of our commonly used flu vaccines, but also as effective. And we're going to produce this in 6 to 12 months. I think that's I mean, that's a tall order. I don't want to be a naysayer. I'm not. I think this stuff is really exciting. The technologies, the imperial vaccine is a technology that, has been around for quite a few years now, but we've never really applied in this way yet. It's been on the back, but it's now being applied, and it's extremely exciting. It's just, will it be safe? Will it be effective? There's also a whole conversation around the fact that we don't really understand the immunology of COVID very well. We're getting a better understanding day by day, but when we really get down to it, that intersection between the patient's underlying factors like age, health conditions, and then what happens in terms of the immune response is incredibly complicated. And unfortunately, I think this is going to be recapitulated when we talk about the vaccines and how
Yes.
effective they are. So I think we're going to have to be very, very cautious. I don't want to say that I wouldn't want to tell somebody or someone in government we'll have a vaccine in 6 to 12 months that's going to be allow us to go back to having everything that was pre-COVID. I don't think that's going to be a reality. Most vaccines take 10 plus years to go from concept to full-scale implementation through the clinical trials. So I think we will have to be patient, but what's been exciting is how rapid we've been able to take some of the technologies we've been developing in the background, been working on applying in the vaccinology field and getting this into potentially an effective COVID vaccine trial. So that is really exciting.
You raised a very good point about the safety requirements, and this is really because a vaccine is given to healthy people, and so you've really got to make sure that you've got that right. I wanted to ask you a little bit about how much of these new vaccines have been based on what we've been learning through infection with other coronaviruses that we've been able to adapt to this particular one.
So there's quite a lot. I mean, the thing is that obviously our prior experience with SARS and MERS has given us a starting point with this pathogen, with COVID-19, that is pretty good. So we understand, we think we understand a bit about how we develop protective immune responses to coronaviruses. And that has been the basis of the selection of the antigens that people have been testing, that sort of thing, has all really come out of what the previous observations with other coronaviruses. However, I would say to you, we have coronaviruses which seasonally transmit through human populations every year, and we don't necessarily develop effective immune responses to those coronaviruses. That means we don't ever get them again. It's a complicated story. And what I am particularly interested in is to see how it plays out is how, when a person becomes infected, because one of the key hallmarks of being able to develop a good vaccine easily is that once a person becomes infected, if you can see that they develop really good, long-lasting immune responses, protective immune responses to that pathogen, that gives you a very good idea, a really good clue that you're going to be able to develop an effective vaccine. We don't necessarily have data supporting that is going to be very, very straightforward in this particular coronavirus. In fact, some of the data that's come out recently indicates that people lose what we are thinking are the protective immune responses in terms of antibodies, that they diminish quite rapidly in people after the infection concludes. But antibodies aren't the whole story. So there's a whole other sort of area around T cells and stuff that people are looking at. So what we need to do is establish more carefully what does protection look like in people that have been infected and recovered. Does it exist? What does it look
So,
like?
so can I ask you? So, with these new vaccines, you've mentioned T cells, and it seems to be in the young, in the young, the T cell response is vigorous, and in the elderly, it's antibodies, and the T cell response is not as vigorous. So, with the vaccines, are people looking at T cells, or are they
So
just looking at antibodies?
no, they're the vaccines that I mean are obviously trying to promote both responses. Blocking antibody has always been seen as a key benchmark for any of these for most of these vaccines, at least the ones that I've been reading about. But you have to have good T cell responses, T helper responses to drive good antibody production. I imagine what we will find out is that either the T effector cells, either in the form of helper cells or maybe even cytotoxic T cells,
Yeah.
are going to be playing a key role. But this is something that's still being explored. We don't really know yet because this pathogen has only just appeared,
Mm.
whatever, 12 months ago, not even 12 months ago. And so our prospects for developing good longitudinal observational trials has been limited. And, we don't really have a lot of good models for these coronaviruses that we can use outside of what we can observe in humans after they become infected.
I think
One
this
of
is an absolute key point in the fact that looking at human immunity, and if you've used a candidate vaccine and you want to see did it actually give protection against infection, you cannot do the challenge because obviously it would be unethical to do that in humans. So you need to follow cohorts through over time and see whether you get a statistically different infection rate in those that have received the vaccine and those that hadn't.
If there are some conditions in which you can do that, Sally, I wouldn't necessarily say that's how this is going to play out. But like, for instance, when people are developing malaria vaccines, they use specific strains of malaria where they can give them to people and then drug cure them very quickly. And so if we had good pharmaceutical agents that could, knock out a coronavirus infection, then you might see
wouldn't need
people,
it.
develop, well,
I
maybe you
think
don't need it anyway, but yeah, exactly.
The
It
challenge.
removes the problem of a vaccine. But I think having a vaccine is going to be a key element in our palette of what we have to have in place to deal with this pathogen long term. I,
David,
I
you
think.
said, sorry,
Sorry.
you say that having a vaccine is going to be key, but one of the main interactions I have, particularly on social media, are people who are, if you like, against the idea of vaccines and very suspicious of the idea of vaccines. So one of the challenges we face as scientists is actually educating the general public that vaccines are safe, and the fact that we're rushing this vaccine development,
Hmm.
it is quite a challenge. So yeah, I think even if we develop a really effective vaccine, would we be able to get enough of the population to agree to take it? Simply because we haven't convinced them that they're safe. And that's a big challenge, just as much as it is to develop the vaccines themselves.
I think that's why it's going to take. I think that besides the science itself,
Yeah.
actually getting something that works,
Mm.
I think that's going to be one of the key things. Um, Vaccines have been very controversial for a couple of for a couple of decades now, and we've seen the results of that in terms of, for instance, measles outbreaks and things like that. Diseases that had disappeared, ostensibly in the UK suddenly are reappearing because people are not feeling safe to take vaccines. And that actually comes back to a couple of points ago where we were talking about intersectionality of the different
Mm.
disciplines
Yeah.
and how I think scientists have to work a little bit more effectively with some of the social scientists and public health promotion experts, and to try and develop the right communication and dissemination processes so that we can help once we do have something that's good and is safe, because that's the key. If it's not safe and we rush it
Yeah.
through, that's going to cause more problems than I think it's going to solve. So I think developing that effective intersectional working process where we develop something and then also work well with other people who help us bring this out to the public. That's going to be a key part of this
Yeah, and
process.
And that's where the
Absolutely.
training of scientists comes into it, that you have to train scientists to be able to communicate in a language that the general public understand, not just the straight scientists talking to scientists.
Yep.
I think it's absolutely key that we get this multidisciplinary approach to tackle this, because there are so many different aspects that have come to the forefront with COVID-19. And
Absolutely.
so I think really it's so crucial that we do work together, and we break down a lot of these disciplinary barriers that are there, and we work with colleagues not only in one country but globally. And I think sort of a lot of questions need to be asked about the global response. What happened to the early warning? What happened to our global preparedness? And these are all things that we need to reflect on once we get on top of this infection, to see how we can improve and how we can go forward.
Yeah.
Because I don't think COVID will be the last. It may be the most recent, but it's not going to be the last. And we had a couple of near misses a few years ago. You know, people always forget. I guess, swine flu was coming around. Everybody thought that was going to be a big issue. And we luckily, at least in the UK, were spared. But other countries, it was a big issue. So I think that was another thing that has made a massive sort of opening people's eyes, because it's here and it's now. It's a pandemic that has come to Britain, and where I came from in the US, people hadn't been accustomed to this sort of thing, and suddenly there's you are part of a global pandemic, and I think that hopefully will focus people more on how are we dealing with health, how are we dealing with our own responses to different events that need not just local or national, but global responses. How are we going to create an integrated environment where we can tackle this as a planet, as a global population, not just as a little, community. And I think that's the key thing.
Yeah, it's a real positive thing in a way that's come out of COVID-19, is the fact that policymakers are now very well aware of the need for healthcare, for science, research, all kinds of things like that. So there are a lot of positive aspects that come out from this.
Well, yes, to a certain extent, it's positive in those countries and those areas where the politicians listen to the scientists, and where the politicians don't listen to the scientists, and the population don't listen to the scientists. Then you get the sort of widespread, if you like, effects of COVID. So definitely you have to have the science training, have the politicians and the population listen to what the scientists are saying.
Yes, and actually, that interface between science and politics has been something that has been very obvious, and not always very not always very successful in the last.
Sorry, one of the hiccups that we had as scientists was in the UK and Europe, was having a UK and Europe centric view about COVID being flu-like, whereas in Asia they'd already had SARS and MERS, and they knew and know now how to respond to it. So our scientists failed in that regard. They didn't take into account previous experience. So it's been a harsh lesson, that one.
I would agree with that. I would agree. And I think there's still some more lessons coming. We're still going to learn a lot. And it's going to be a hard road in some respects, but I think it's also an exciting opportunity for scientists, and I think our young scientists, to make their mark, and to really contribute positively by being part of these different aspects of development, either vaccines, drugs, intersectional study with COVID and healthcare provision, all of this stuff, diagnostic, all of this stuff, technology development, amazing opportunities. And I think for me, I would find it a very stimulating time to kind of come out of a degree and say, okay, where do I go next? Well, there's all these different potential places. and they're all very exciting. I'm just going to draw us to a conclusion. I want to thank all of you. I mean, this has been such a great conversation. I certainly have learned a lot and really valued listening to some of these things, and I hope the people who watch this will also enjoy it in a similar way. I want to thank Professor Seed, Professor Cutler, and Dr. Morgan for their time.
Thank you.
Thank you,
And and to all of our students who are returning to campus, we're going to see you soon, and hope we're going to be talking a lot about COVID. So I hope you aren't tired of it yet, because we're going to be doing a lot of COVID talking when you come back. Because it is such an exciting subject to put that's right in your life right now. All right, thank you very much, guys. I
Thank you.
appreciate everything.
Take care.
Thank
Bye
you.
bye.
Bye bye.
Okay, bye. Nice to see you. Bye.